This video is part 2 of Chapter 3, Sensation and Perception for PSY 110S, the Psychological Sciences. In this section of the chapter, we're going to be looking into our dominant sensory systems, vision and hearing. We're going to start with thinking about the characteristics of both light and sound, because these are the attributes that psychophysicists are going to measure as they try to get some parameters on what we can see and what we can't see, what we can hear, what we can't hear, and so on. So we start with wavelength, and that's the distance in one cycle of a wave from one crest to the next. With respect to vision, people can see between 400 and 700 nanometers. Now, right now, that doesn't mean anything to you. Just understand that depending on the species and how that species has evolved, their vision is going to be unique to their needs. We need to be able to see in this particular range. Amplitude refers to the amount of energy, the strength, the intensity of the wave. It's going to show up visually on a screen as the height of a wave at its crest. For light waves, amplitude is related to brightness. Frequency is something we use to describe auditory stimuli, so in hearing. It involves the number of times the sound wave cycles in 1 second. Shorter wavelengths reflect higher frequencies, so higher tones. Broader wavelengths are related to lower tones. This determines pitch. So like the difference between, if you're looking at a piano keyboard, far to the left would be the lowest tones, far to the right would be the highest tones. Those higher tones are going to have higher frequencies. The lower tones are going to have lower frequencies. As with vision, our hearing is really bounded by our specific needs, with the greatest degree of sensitivity to sounds that are at the level of human speech. Now, to give you a visual of what we're talking about here, wavelength goes from that peak to peak. Amplitude is height, and that will be different depending on what kind of visual stimulus is coming into the eye, for example. And then for frequency, for hearing, you've got low frequency waves, which are lower pitch, and high frequency waves, which are higher pitch. Now, our sensory systems obviously need to interact. We think about the way when you're eating food, it's a full-on sensory experience. You see the food, you smell the food, you hear aspects of the food and you yourself eating. You feel the textures of the food in your mouth. All of those sensory experiences are going to, in some ways, mix. Now, for some people, people are able, they have some crossing over of their sensory systems. I hate to use the word normal, in regular everyday people, in other words, people who don't have synesthesia, we just perceive our senses as, sometimes interacting. The experience of eating food is a really good one there, that you have, smell, taste, touch, vision, and even hearing that are involved in all of those things. Synesthesia is a rare condition. There are many famous people who have come forward and described themselves as synesthetes. This neurological condition involves having cross-sensory experiences. So a common example is having auditory signals be perceived in terms of color or even shapes. That and vice versa, hearing certain tones and seeing color, seeing certain images and hearing certain tones. One particular example of this, Tori Amos is a singer and songwriter. She is also someone with synesthesia. She processes sounds in image form. So she hears songs, or hears tones, hears, differences in pitch, but she sees things that she calls light creatures. She has never seen the same one twice, but those images become very alive as she hears different sounds. In general, synesthesia or not, the brain is going to process incoming sensory information. The brain can't process just the physical energies, so they have to be transduced. Transduction refers to this way in which we have to convert the physical energy, like light waves and sound waves. They have to be converted into neural signals by specialized cells within our sensory organs. This conversion process is called transduction. Now, for a little bit more about synesthesia, I have this video clip that I found on YouTube, so I'll play it and then we can talk about it.
If you try to move your eyes smoothly from left to right, you can only do that if you see a moving object. So when you go to a doctor's office and the doctor tells you to follow my finger, they're doing that to see your smooth eye movements.
And so we set about setting up an experiment in my lab which had eye tracking and motion tracking systems.
And the question we asked was, the people who report seeing their own hand in darkness, are those the same people that give us smooth eye movements? And the answer was yes.
The eye tracker uses infrared cameras, so it illuminates the eye with infrared light, which is of course not visible to the eye. So you can track an eye in complete darkness.
So to us, this was a way of being certain that our subjects are experiencing motion, because if they didn't see any moving object, they wouldn't be able to follow this smoothly. And it varies from person to person. For some people, they don't see anything at all. For some people, this thing is very, very vivid.
I had a visual perception, even though it was completely dark. It was like a very faint blob. It was more motion than anything, and I could follow it.
What we have in this experiment is really a combination of two different senses. We have a kinesthetic sense that's aware of where your hand motion is. We have a visual sense, and they're clearly talking to each other. And this led us to synesthesia because synesthesia is a condition that's characterized by sensory crosstalk.
My strongest synesthetic associations are with graphemes, which are letters and numbers and symbols, and color. So when I look at a word, each of the letters have a specific color that I associate with them. I don't physically see the color, but in the back of my mind, I have those colors sort of floating around in there.
Maybe people who have higher connectivity in their brains will actually experience this to be much, much stronger. And the result was the synesthetes in a study were completely off the chart. And in some cases, when we measure the eye movements of the synesthete in just a lit room like this one and in complete darkness were identical. You couldn't tell a difference. And this never happened for our typical subjects.
The standard model of sensory processing in the brain is that sensory signals from different modalities like hearing and sight and touch are all processed by independent brain systems. What this study shows is that, in fact, that model's not quite right, that these different sensory areas of the brain are actually communicating with each other at a very early stage of processing.
Synesthesia is like the tip of the iceberg, where it shows that it is possible to take two things that are seemingly so different and merge them together.
It really does make sense, because my audition, my touch system, my visual system are all looking at the same world. So it makes sense they are sort of working together to give me the best perception that I can get.
A production of the University of Rochester.
So in that video, what they're really trying to get across is that all of us have interactions among our various sensory systems. What's different in synesthesia is that there appear to be more cross connections that create these unique experiences that synesthetes have. So shifting gears a bit, we're going to look specifically at how this transduction process works in vision. So we have to look at, what is the pathway that light travels as it goes through the eye and then eventually to the brain for processing. This image from your textbook shows first how light waves are going to be transduced by what are called rods and cones. Those are the neurons that are specifically designed to deal with light stimuli. So the neural impulses, they'll be transduced by the rods and cones, which means that they're taking the raw light information and converting it into a neural message. Those neural impulses are then going to be sent on forward for eventually to end up in the occipital lobe. The rods and cones send that image information to the bipolar cells, and those then pass them through another layer of cells called ganglion cells, and those are shown in the image as with the dark arrows. And then the axons from the ganglion cells are all bundled together, and eventually that combined group of cells becomes what's called the optic nerve. And that nerve, which is really a bundle of axons, is going to send the message on, providing visual information about the image to the visual cortex in the occipital lobe, going through the thalamus along the way. So as you're looking at this cross-section of the eye, the things you need to look at, you have a lens, a pupil, iris, cornea, and retina. There's a fovea area and an optic nerve. And in that space, there's a blind spot. So the space where the optic nerve exits the back of the eye, we don't have any perceptual ability. We don't have any ability to sense information. That blind spot is essentially if your eyes were completely dead still, that blind spot would become something that you could detect. Light waves are reflected in the white arrows on the graphic. Those light waves are transduced, as I said before, by the rods and cones at the back of the retina, generating neural impulses that replicate the visual images. The rods and cones are going to send then their messages to the bipolar cells, which then move them on to the ganglion cells. The ganglion cells, their axons, become the bundle of things that we refer to as the optic nerve. The message then goes through the thalamus to the occipital lobe. So I have an image here, or a video here, just to walk you through this. I learn better if I see things in motion.
Hi everyone, welcome to 10-Minute Neuroscience. In this installment, I'll be covering the pathway of visual information through the brain, starting with the eye and ending with the visual cortex and surrounding areas. This will be a very general overview, and I'll be focusing more on the pathway of visual information than on the processing of that information. But this should serve as a good introduction to the way visual information travels from the eye through the brain. Of course, vision begins with the eye, but the neural aspect of vision really starts with the retina, which is the neural structure of the eye and is outlined in blue here. Other components of the eye, however, help to create a focused image on the retina. This is accomplished to a large degree through the actions of the cornea and the lens. The cornea is a transparent layer at the front of the eye that lets light into the eye. It also bends or refracts those light rays to direct the light onto the retina. The lens also helps to direct light on the retina. It has less refractive power than the cornea, but it has an advantage in that its shape can be modified by muscles in the eye called ciliary muscles. And changing the shape of the lens can help to maintain focus for objects that are closer or farther away. As people get older, their lenses become less flexible and less capable of changing shape to focus on nearby objects. This is why people tend to need reading glasses when they get older. The size of the pupil, the opening in the middle of the iris, which is the colored part of your eye, can be adjusted to regulate the amount of light that reaches reaches the retina. For example, there are muscles in the iris that cause the pupil to dilate or open up more in low light situations and constrict in an environment with higher levels of illumination, since it doesn't need to let in as much light in that type of environment. The retina is a neural structure. It's actually considered part of the central nervous system. Its main function is to detect light and use that light to produce electrical and chemical signals that the rest of the nervous system can understand. You can see here a close-up of a section of the retina. And although there are over 1,000 different types of neurons in the nervous system, there's only 5 basic types of neurons in the retina, and those neurons are situated in distinct layers. So even though there are hundreds of millions of neurons in the retina, compared to the rest of the nervous system, it's relatively simple anatomically, and this has helped us to develop a better understanding of vision than of any other sensory system. Surprisingly, the layer of the retina that's at the very back of the eye is the layer that contains photoreceptors, the cells responsible for converting light energy into electrochemical signals, a process known as phototransduction. The location of the photoreceptors is surprising because it seems counterintuitive to have the light-detecting cells at the very back of the retina. So light has to travel through the eye and then through several layers of cells to reach the photoreceptors. But it's thought that their location is strategic in the sense that they're next to a layer of the retina called the pigment epithelium. And the cells of the pigment epithelium help to maintain photoreceptor cells and keep them functioning properly. There are 2 main types of photoreceptor cells, rods and cones, which are named for their shape, as you can see here. These photoreceptor cells are the site where vision really begins. They each contain hundreds of discs that are capable of absorbing photons of light, and absorbing photons causes the photoreceptors to change levels of neurotransmitter release in order to convey information about a visual scene. Rods and cones have different functional specializations. They're involved in distinct aspects of vision. First off, cones enable us to see color, while rods don't provide for color perception. Rods are also very sensitive to light and have low spatial resolution, meaning they're not good at seeing details. Cones, on the other hand, are not very sensitive to light and have high spatial resolution, so they provide us with higher visual acuity. In low light conditions, only rods are activated. This makes it more difficult for us to make out details when there's very little light, due to the poor spatial resolution of rods, and it also means that in dim light we can't perceive color. When levels of illumination increase, eventually rods stop responding and fail to convey information. Essentially, their high sensitivity to light causes them to become overstimulated, or saturated as it's often called, in normal lighting situations like sunlight or even typical indoor lighting. So in those conditions, cones are the dominant photoreceptor in determining how we see. Surprisingly, even though cones mediate perception in typical light situations, we have far more rods than cones in the retina. There's somewhere around 90 million rods and only about 4.5 million cones. However, there is one part of the retina, an area called the fovea, where there are many more cones than rods. In fact, at the very center of the fovea, which is called the fovea, there are no rods at all. Because of its high cone content, the fovea is the part of our retina that has the capacity for our highest acuity vision. This causes us to unconsciously move our eyes so that important visual information lands on our fovea, since this is the part of our eye most capable of discerning important details. When photoreceptors absorb photons, it causes changes in the amount of neurotransmitters these cells release, and this affects the activity of the next layer of cells, bipolar cells. Bipolar cells pass on signals about perceived light to the next layer of cells, which are called ganglion cells, and these cells will carry the visual information to the brain. We'll talk more about that in a moment, but I want to mention the other 2 major cell types in the retina, horizontal cells and amacrine cells. Horizontal cells have dendrites that spread horizontally and make contact with multiple photoreceptor cells. Horizontal cells modulate the function of photoreceptor cells to do things like enhance contrast and adapt to changes in lighting conditions, among other things. Amacrine cells make contact with bipolar cells, ganglion cells, and other amacrine cells, and they also have a number of functions, but like horizontal cells, they're generally thought to be involved with refining the visual signal through their ability to modify the functions of other retinal cells. So horizontal and amacrine cells are involved with very early processing of visual information, but most of the visual processing occurs in the brain, so the information from the retina has to be carried out of the eye into the brain. This is accomplished by the ganglion cells, whose axons leave the eye in a bundle at a region called the optic disc. Because the optic disc is the area where the ganglion cells leave the eye, and essentially they need a place where they can exit the eye, there are no photoreceptors there. So this creates a small region where we don't receive any visual information or a blind spot. Amazingly, we don't notice this blind spot in our visual scene because the brain fills it in with information from other photoreceptors. If you find it hard to believe that you've got a blind spot always present in your field of vision, Click on this link above for a quick and simple experiment that proves the existence of your blind spot. The ganglion cells leaving the eye form the optic nerve, which is one of our cranial nerves. The optic nerve extends back to this region just below the hypothalamus called the optic chiasm. At the optic chiasm, about 60% of the axons from the optic nerve cross over to the other side of the brain, while the rest stay on the side they originated on. The fibers that are coming from the nasal part of the retina, the part closer to the nose, cross over to the other side, or decussate, while the fibers from the temporal part of the retina, the part closer to the temple, do not cross over. The result of this is that all the information from the right visual field ends up traveling to the left side of the brain, and vice versa. After the optic chiasm, the visual fibers are no longer called the optic nerve. They're now called the optic tract. The optic tracts extend to multiple areas. For example, some of the fibers go to an area in the brainstem called the pretectum, which is involved in a number of visual functions, such as the pupillary light reflex, which causes your pupils to constrict when there's greater illumination in your environment. Other fibers go to a region of the hypothalamus called the suprachiasmatic nucleus. This nucleus helps to maintain circadian or daily rhythms and uses information about light in the environment to help to do that. And still other fibers go to the superior colliculus, a region in the brainstem that, among other things, helps to coordinate head and eye movements to focus on objects of interest in the visual field. Most of the fibers in the optic tracts, however, end in a nucleus in the thalamus called the lateral geniculate nucleus. There's one on each side of the brain here. Here, the optic tracts synapse on neurons that leave the lateral geniculate nucleus and extend toward the back of the brain as bundles of fibers called the optic radiations. The optic radiations travel back to a region of cortex that surrounds a fissure called the calcarine sulcus. This small area of cortex that surrounds this calcarine sulcus is called the primary visual cortex, or V1. It's represented by this striped area here. There's a collection of myelinated fibers that forms a white stripe here that can be seen with the naked eye in anatomical brain sections of this region. And because of this striation or stripe, sometimes the primary visual cortex is called the striate cortex. The primary visual cortex helps to make a visual image out of the information that has been received by the retina. To do that, there are neurons in the primary visual cortex that are activated preferentially by different characteristics of a visual stimulus, such as orientation, movement, contrast, depth, etc. The primary visual cortex also communicates with a multitude of other visual areas that surround it, including visual area 2 or V2, visual area 3 or V3, V4, V5, and V6. Neurons in these other areas seem to be specialized to some degree for detecting specific aspects of a visual scene. Neurons in V5, for example, seem to be specialized for detecting movement. These additional visual areas are recruited by V1, and they also begin to recruit other areas of the brain to accomplish higher level processing of visual information. This enables our brain to move from identifying the basic aspects of a visual scene, such as shape and contrast, to more complex tasks like object recognition, which can provide the image in our brain with meaning based on previous experience. So we've just scratched the surface there, but I hope that gave you a sense of the major pathway that visual information travels in the brain. Thanks for watching.
He does so much better of a job than me in describing that. If you enjoyed that video, 10-minute neuroscience with this YouTuber, Neuroscientifically Challenged, is well worth your time and interest. So just to review what he described there in the video, you don't have any visual processing that happens until the light waves actually reach the retina. That light is coming in and then it reaches those sensory cells, the light-sensitive layer of the eye that has the ganglion, bipolar, and receptor cells, the rods and cones. The light waves pass through the ganglion and bipolar cells before they reach those sensory neurons, and that's when processing actually starts. The light waves are absorbed by photopigments, so these are light-sensitive chemicals inside the receptor cells. That's what creates a pattern of neural impulses. It's the pattern that arrives at, in the brain that then is interpreted and made sense of. Neural information, once it goes to the bipolar cells, it's then sent along to the ganglion cells and then across the optic nerve and as was described in the video that you just watched, there's some crossing over and duplication, which is all really essential to how the brain makes sense of the visual stimuli that are coming in. The brain is designed to make sense of visual stimuli by breaking it down into its features. So, excuse me, imagine that you're looking at, say, a triangle. The brain is going to not immediately, not right off the bat, identify, oh, triangle. It's going to break it down into features, so angles, straight lines, but then they're going to be regrouped so that you can put them together and then recognize, and eventually, with further processing, you're going to use language to name it. So to recognize a triangle, it's broken down into features of two straight lines at different angles and a horizontal line. Then they're recognized when they're regrouped in a proper orientation and in relationship to one another. There are cortical cells that are basically their entire job is to detect certain kinds of features. So, say in a triangle, the angles that the upward lines are at will be perceived by specific cells that are there. They have the job of looking at those kinds of lines. Then there are other cells that their specific job is to detect horizontal lines, and still other cells that would be detecting perpendicular lines as opposed to lines at an angle. These elementary features then have to be combined, and that's a higher level processing issue where you're taking the whole and understanding the interrelationships among the features. So recognition of an object involves taking things apart, essentially, and then putting them back together again such that they can be identified. One of the most important objects that we are designed to perceive is the face. So we're born with a capacity to detect and perceive features that, when they are in the proper orientation to one another, definitely appear face-like. And once we see those objects, it's very hard to avoid perceiving them as being faces. There are separate processing systems for such things as forms, so different shapes. There are processing systems for color, for distance, for motion. Now the brain is going to do the heavy lifting of coordinating and kind of like you have different instruments in an orchestra, you have to pull all those sounds together to produce the one thing. Our visual system does the same thing. You have all of these elements of what we see, color, shape, size, motion, distance, and then they have to be coordinated so that we can use all the information simultaneously. Now let's take a look at how feature detection has been studied, and I hopefully it will kind of help you wrap your head around this idea of feature detection.
David Hubel of Harvard won a Nobel Prize with his colleague Torsten Wiesel for mapping the action of receptor cells along the visual path of a primate, from the retina to the cortex.
Receptor cells usually in the visual pathway mean the cells that take in the energy into electrical signals. And in the retina, those are the rods and cones. There are 125 million rods and cones in each retina. So in the case of the visual pathway, you start with the retina. The output is the optic nerve, which contains 1 million fibers. They end up in a certain region in the brain. That region, or there are really 2 or 3 regions that they end up in. Each of those sends a cable of fibers of the order of maybe 1 million to other regions, and they connect to other regions. And in the case of the cortex, you have separate areas of cortex, each one connected to one or more other areas. And this whole thing is a pathway. In the cortex, for example, the primary visual cortex, which is about 7 stages beyond the receptors in the retina, those cells react to visual stimuli only if a line falls on the retina, and the line has to be a particular orientation. It can be a bright line or a dark line or an edge between bright and light. Any kind of line really generally works, but the position of the line and the orientation are terribly important, and if they're not just right, any individual cell doesn't respond.
In this experimental footage, a bright vertical line stimulates a small number of neurons in the visual cortex of a cat. The crackling sound is the electrical activity of these neurons as they respond to this retinal image. By listening to the intensity of this electrical activity, the researchers can determine the correct orientation of the line. But when the line is moved to a diagonal or horizontal position, the amount of stimulation decreases dramatically.
We're only at a very elementary stage when it comes to understanding something like how you recognize a face or something like that. The general region of the brain is known where things like that go on, but we don't have the slightest idea of what's happening at the level of single cells for that particular problem. But for more elementary problems, for the very first processes of vision, we do have a very good understanding of what happens at the very beginning. So it's just a start.
Misha Pavel of Stanford University is studying the successive stages of information processing that take place continually as we perceive the world. Using computer graphics, he has demonstrated how the visual system breaks down visual stimulation into millions of bits of information and recombines them into a coherent image that we recognize.
Seeing something seems such an effortless activity that it's hard to imagine for us the complexities and difficulties that are involved. Only when you try to build a robot that can actually see and recognize objects do you realize how complicated task this is. People must do an awful lot of processing in order to see images and interpret them.
Here is an image of a cat as our eye and brain sees it. Now let's look at what the visual system goes through to enable us to see this image. When the light first reaches the retina, the image is slightly defocused by the optics of the eye. Then it is broken up into millions of little pieces. Each receptor sees just a minuscule portion of the original image and measures its brightness. Different receptors in the retina are sensitive to different colors of light and respond to the amount of that color that they see. To discover objects, the visual system tries to find important boundaries. It uses edge and line detecting neurons, whose characteristics have been investigated by Hubel and Wiesel. Here, you can see the results of the red, green, and blue edge detectors. Look at the blue edges. It is hard to believe that there is a cat in there.
What I'd like to show you now is the kind of information that the brain uses in order to make sense out of these messy images.
The things that the brain seems to look for are constancies and simplicity. Imagine that you are in your visual system looking at this pattern of active neurons. But you can't recognize the pattern. We can see it now because all the points in the cat picture move together. This is an example of the brain using rigidity to recognize moving objects. Ambiguous perception of motion can actually destroy the rigidity percept. In this case, we have a rotating rigid object, a square, but when its corners disappear from view, the square appears to get smaller. When the corners reappear, it gets larger. Another example. We thought that the square loses its rigidity because the visual neurons at each location can see only a small proportion of the entire picture and therefore can't accurately perceive the direction of the moving parts of the object. If we rotate the cross, then the stationary square appears to be rigid. We can simultaneously compare these two situations. The conclusion is that the motion of the square is necessary to lose rigidity. The failure of rigidity in this case helps scientists to study how information from different retinal locations is combined to form a single percept. This is one of many examples where perceptual phenomena can reveal how our brain works.
So part of what this helps us to understand is why is it so hard to get a self-driving vehicle? What we do with our perceptual system, the level of feature detection alone is incredibly complex, but also very fluid. And then the later processing of that information to give it meaning, those are some of the things that we are just not there yet to be able to program a computer to do in the way that we do it or as fast as we do it. And that concludes part 2 of chapter 3.